So is the screen visible to all?
No, ma'am.
No. Is it visible now?
Yes, ma'am.
So, what is genomic library? Basically, it represents a complete genome in multiple clones, each containing small DNA fragments. So, what happens is like your genome is like very large. If we talk about human genome, right? And if you want to know the sequence of some gene, so what you do is you make multiple clones containing partially digested fragments, and then you sequence it. So to know the complete sequence of particular gene, you can make many clones containing those small fragments, and then do the sequencing, so that you can know the exact what is the sequence of any particular gene which was not known earlier. So basically, in the starting, we'll be talking about the introduction part of what is genomic DNA library. Then we'll be covering the steps include to form this complete library preparation.
A library means a collection of books. So a genomic DNA library would simply mean a collection of genomic DNA, right? Okay, so these days it is high throughput sequencing era where we can in fraction of seconds.
So this audio is completely audible to everyone, right? The audio which I am playing in the introduction part.
Yes, ma'am. It
Yes,
is
ma'am.
Okay,
clearly
okay then.
audible.
2 minutes we can sequence a full genome. But let me take you to far 70s where sequencing was not so much developed, but people were eager to sequence the genome of yeast, bacteria, virus, and even human. But there was a huge challenge in this work because the genomic size. A mammalian genome would be in order of 10 to the power 9 base pairs. So big, right? Now, there are three basic problems. How to clone such a big genome? Then, where to clone it? What are the appropriate vectors which would be useful for cloning such a huge quantity? And then, even if we clone it successfully, then how we can identify our sequence of interest from this library of the sequence. So screening of this library was another challenge. Now scientists were always bothered that okay, if we try to approach this problem, we should first understand what vector to use. Second, we should understand that even if the cloning is successful, we should devise a efficient strategy. And before that, we should have an estimate that how many recombinant colonies we have to screen. So these were the challenges at that era. Now, human genome, if we talk about the human genome, which is like somewhere around 2.8 into 10 to the power 6 kilobase pairs, and we desired to clone it, I wanted to appreciate that how difficult it would be. Because let's say we want to clone it and we would restriction digest this whole genome, and and let's for a moment assume that we are going to clone it inside a bacteriophage. And the insert size, the insert of our fragment that we are going to clone would be 20 kb. In order to cover this whole genome with such 20 kb fragments, we would at least need 2.8 into 10 to the power 6 kb divided by 20, which is approx 1.4 into 10 to the power 5 fragments. At least this number of recombinants we have to uh, screen. But imagine the number is more than that because sometimes some particular fragment would be overrepresented and some fragments would be underrepresented. So we have to, in order to be safe and get the whole coverage, we need to screen more colonies. Now it's still difficult. You can understand how challenging it is mathematically to imagine how many colonies to screen. Now Clark and Carbon came up with a formula. The formula is very looks very simple yet elegant. So they calculated number of individual recombinants to be screened based on a probability. So they defined a probability P, which is including a DNA fragment in a random library. And they came up with this formula. Now, if we consider we want to screen the human genomic library with 95% probability, and provided our insert size was 20 kilobase pair, then plugging in all the values to this formula gives us at least we need 4.2 into 10 to the power 5 number of recombinant colonies to be screened, which was the number pretty big that we got earlier. So minimum number was like 1.4 into 10 to the power 5. Now the number is 4 times more than the minimum number, right? Now if we want to have a probability which is 99%, which we are becoming more stringent towards the coverage, we have to screen even more colonies. And screening 6.5 or 4.2 10 to the power 5 colonies is not a joke. So it was damn challenging at that time. Next question is, what vector should be appropriate to make a genomic DNA library? So there are two options. One option was lambda fudge. Lambda fudge based vectors are easy to screen. Thousands of lambda fudge vectors could be screened simultaneously, and their transformation efficiency is pretty high, even higher than the bacteria. In comparison to that, the carrying capacity of bacterial artificial chromosome is pretty high than these lambda fudge vectors. So bigger fragments, cloning bigger fragments is easier in back. So both the approaches could be taken to clone the genomic DNA library. Now in lambda fudge vector, so their genome is 49 kilobase pair. And few interesting facts about their genome is 1/3 of their genome is just trash. They don't need that. So instead of that particular genomic region, it could be replaced with our gene of interest or the DNA sequence of interest. So we can cut this tougher fragment out and we can insert our DNA of interest. And we can have several thousand plaques created by these lambda fudge vectors. But now question comes, which restriction enzyme to use? Imagine we use a 4-base cutter or a 6-base cutter, only one type of restriction enzyme. So only one type of restriction fragment would be generated. Only one type of length would be generated with no overlap with the previous one. Is that advantageous? There are also other options where we use two restriction enzymes, let's say one 4-base cutter and one 6-base cutter in combination, and we do a partial digestion, which would give rise to several different random fragment length, but each of them has a probability that it would overlap with some other fragments. So there could be two strategies, but which would be useful? Imagine if you have a puzzle and you know that puzzle gives rise to a picture, and you get some amount of the information of the previous block, it's easy for you to solve the puzzle. Now if you don't have any overlap between the other puzzle blocks, it would be a lot more difficult to solve the puzzle, right? So scientists always wanted to have more overlap such that they had more clue and they don't get lost when they're screening the libraries. So that's why nowadays people use a combination of restriction enzymes and do a partial digest for cloning. Now after the cloning, when you get the recombinant colonies on Petri plates, you can take them out and you can screen them, sequence them, and you get multiple different sequences of different lengths, definitely, right? Now you know that these sequences have some degree of overlap with each other that you can call contig, and then keeping one particular landmark and arranging all these contig on one top of another, you can retrieve the physical map of the genome. And that is how the scientists solved the this genomic library problem, and they really sequenced the genome like this. So that is how genomic DNA library was pretty useful at that particular point of time. Now there are other type of libraries like cDNA libraries, which I had a different video of. But cDNA libraries are different. Genomic DNA library would contain all the sequence including introns, exon, non-coding, repetitive sequence, everything. But cDNA libraries would only have the the sequence of exons which are expressed. Now, cDNA library would only have some sequence of those genes which are expressed at that point of time. But genomic DNA library would have the sequence information for all the genes which are present in that organism at any given point of time.
So now you know the basic of what is genomic library and maybe few things like what you need to prepare a genomic library, right? But there are few steps which needed to be covered to prepare a library. First of all, supposedly you want first of all you have to get a genome, and for that you need to isolate genome. Suppose you are interested in preparing a genomic library of some mammalian cells, so you'll take some mammalian cell and isolate genomic DNA from that particular organism. Suppose you are interested in preparing this genomic DNA library from other source of organism. So depending on the organism we take, the method of isolation of genomic DNA varies, because like cell wall, the if you see the organization of any particular cell of any organism, then you'll be like supposedly, just an example, plant cell, yeast cells, they have cell wall, right? So cell wall is like not easy to break. So we'll be isolating the genomic DNA in those particular organisms using some other method. And suppose for mammalian cells we do not have cell wall, so we just have cell membrane, which is easily breakable. So we can choose some different method of lysis of cells for that particular organism. Then once you have isolated complete genomic DNA from the organism of interest, then you need to cleave using restriction enzymes. So cleavage of genomic DNA using restriction enzyme, it depends on your interest too. Like in the introductory part, they try to tell ki either we can use 4 base pair cutter and 6 base pair cutter, or either we can partially digest it with both the like 4 base pair and 6 base pair cutters. So this cutting with both the 4 base pair and 6 base pair give us an advantage of having overlaps. I mean, we'll get a sequence which is overlapping with each other at a particular point, so we can organize the sequence key. Okay, this is overlapping, so it must be coming in continuation with that particular with the previous sequence, which which give us the possible direction of sequence. Then third is, yeah.
Ma'am, just the sorry for the interruption, ma'am. Just can you explain the third step, ma'am? I didn't get that. The digestion of the vector and CIAP
So
treatment.
I'm going to come to the third part. Actually, I was explaining the second one. Digestion of genomic DNA I explained. I'm coming to the third part. So third part is, suppose you have gene of interest now, but now you have to clone it in a particular interest of vector. So depending on the vector you have chosen for your cloning, you can choose different vector according to the size you want to clone. Suppose you have a gene of interest having larger base pair, so you need a vector which can accommodate that particular length of gene of interest. So you'll digest the vector. So like majorly most of the vectors have this MCS region called multiple cloning sequence sites, where there are sequence of many restriction enzymes. These enzymes can bind to these sites and cut them. Okay? So you will be digesting the vector, and you'll give CIAP treatment, that is alkaline phosphatase treatment, which removes the phosphate group from the digested vector, so that the vector cannot self ligate to itself. Then the next step is to suppose you have a digested vector. Now you have your gene of interest. Then now you will ligate the fragmented DNA with the vector, and then you'll transform recombinant DNA into bacterial cell, and then screen the transformed DNA. So each step will be reading now how to do it and what procedure you can follow up. So first is isolation of genomic DNA. So suppose in this example, I'll be showing you how to isolate gene from E cell. So I told you this thing, E cell has a cell wall, right, which is made up of chitin. So we need some stronger lysis reagent which can, easily break the cell wall and cell membrane. And suppose in mammalian cell, you don't need that much of, detergent, because mammalian cell doesn't have cell wall to it. It just has cell membrane, which is easy to break. So for E-cells, we usually take CTAB method, which is a detergent which binds to cell wall and breaks it. So to isolate this genomic DNA, first you will lyse the cell wall and cell membrane, and then you will extract. So once you have lysed, you have RNA, protein, DNA to your cell. So we will be separating protein, DNA, RNA from each other. So we uses chloroform phenol extraction, chloroform phenol isoamyl alcohol extraction method we call it. So I'm going to play a video which shows how to do it. Is this video visible to all and the audio is clear?
Ma'am, no audio is there.
Oh, let me just again wait.
Ma'am, can you make it a full screen if possible?
Let me just see it once. Can you see it's like stopped? Is it audible now?
Yes, ma'am.
Yes.
Okay, actually I cannot make full screen to the video. Sorry for that.
Okay,
Uh,
ma'am. No problem.
I'll just increase and so we have added RNase A. It is to degrade the RNA. So once we have degraded the RNA, what we are left with is protein and DNA, right? So further we'll be isolating or separating the DNA part from the protein part using chloroform and phenol. And this proteinase K is for degrading the protein. So this chloroform and phenol, what it does, it makes the solution into two phase. One is aqueous phase, one is organic phase. So your aqueous phase contain the DNA, whereas your organic phase contain all the protein part. So it's written like you just take the aqueous phase, which contains all the DNA material to it. So as I've told that chloroform and this phenol, what it does, it separate the aqueous and organic phase. Okay. So once you have taken the aqueous phase, it contains all the DNA. Now you need to precipitate the DNA. For that you add ethanol to it. So 100% ethanol precipitates the DNA, and then you just air dry or like take off the supernatant, and your pelleted DNA is your genomic DNA, which you dissolve in TE buffer. And then you this, like, supposed to now once you have isolated the DNA, you need to analyze it by running on 0.8% agarose gel, and then you measure the concentration of that DNA using maybe nano drop or some other methods like plate reader or anything else, like on you can measure the optical density of your DNA. So once you have the genomic DNA, the other thing you need to have is the vector. So the selection of vector depends on the size of interest to be ligated. So depending on the size, we can have different vector. We can choose different vector. So next step is to cut the restriction, like to cut the vector as well as your genomic DNA. with multiple restriction enzymes so that you can have this linearized vector and linearized various fragments of your genomic DNA. So in like when we used to have these experiments in UG lab, then we used to use PUC19 vector, okay, and we used to cut it using SMA1 enzyme to make blunt-ended linear vector. Next is digestion step. Like both the vector and in genomic DNA, you uses the same procedure. Like the digestion procedure is same for both. So for digestion, I'll be playing the video.
Materials required: 5 microliters of deionized water, 1 microliter of 10x reaction buffer, 3 microliters of plasmid DNA, zero point 0.5 microliter of eco-R1 enzyme, zero point 0.5 microliter of
These enzymes used are in this video are just for reference purpose, okay? And the enzyme you guys can use uh, 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 depends on the vector you use, and depends on the 4 base pair cutter you need, 6 base pair cutter you need for your genomic DNA fragments. It depends on the experiment you are performing.
BAM-H1 enzyme, centrifuge, water bath, ice box, floater, empty vial labeled vial 1, waste beaker, pipette and tip box. Procedure: take a 10 microliter micropipette and adjust to 5 microliter. Insert a fresh micropipette tip into it and transfer 5 microliter of deionized water to an empty vial labeled vial 1. Similarly, add 1 microliter of 10x reaction buffer, 3 microliters of plasmid DNA, zero point 0.5 microliter of eco-R1 enzyme, and 0.5 microliter of BAM-H1 enzyme to the same vial. Mix the contents of vial 1 thoroughly and spin the contents in a centrifuge for a few seconds. Next, place the vial in a floater and incubate the mixture in a water bath at 37 degrees Celsius for 1 hour. Store the vial at -4 degrees Celsius in the freezer after incubation. Precautions: make sure that the restriction enzyme does not exceed more than 10% of the total reaction volume.
So now each enzyme we use for digestion of vector in genomic DNA, it has some specific, like each enzyme has unit to it, like activity to it. So depending on the concentration of enzyme we are using for a reaction, we can get larger fragment, medium fragments, and smaller fragments. For just an example, it's not specific to any enzyme. It's just a hypothetical example. Suppose you use 1 unit enzyme per 250 microliter water. That means it is like lower concentration of enzyme, right? So you can get larger fragments by using that particular concentration of enzyme. And if you make the enzyme more concentrated, suppose 1 unit per 50 microliter of water, since the concentration of enzyme is so much, it can give you smaller fragments. And the same depending on the time we are using. Supposedly, you are using the enzyme, like you are putting the digestion for a larger period of time, then you can get multiple smaller fragments as compared to suppose you are just taking out the reaction in just 1 hour or so, then you can get medium to larger fragments from it. Okay, once you got the fragments, like once it is digested, we need to purify the digested product in your vector, because since digestion buffer has many salts to it, we need to have those fragments, like we need to separate those salts from your ligated sorry digested product and vector. So you need to run the digested vector as well as your gene of interest on agarose gel, and then column purify the fragments of desired length. Okay, then you need to give CIAP treatment to your vector. CIAP is alkaline phosphatase. What it does, it hydrolyzes the 5 phosphate group from the DNA. It is basically done to remove the 5 dash phosphate group so that vector cannot self ligate to itself.
Dephosphorylation can be an important step in a cloning experiment, as it helps to prevent self-ligation of the vector. In the cloning workflow, inserts and vectors that have been generated by restriction enzyme digestion possess a 5' phosphate and a 3' hydroxyl group, which are necessary for ligation to proceed. When working with vectors that have been digested with a single restriction enzyme or 2 restriction enzymes with compatible ends, removal of the phosphate group will prevent vector self-ligation, reducing the background of transformants, which do not contain inserts. This will decrease the number of transformants needed to screen for clones with the correct insert. If your insert is a PCR product, it will not have 5' phosphates unless specified during primer synthesis or added by T4 polynucleotide kinase. For optimal cloning results, we recommend ligating a phosphorylated PCR product to a dephosphorylated vector to minimize self-ligation of vector and reduce background colonies. The dephosphorylation reaction requires the hydrolysis of the ester bond between the terminal 5' nucleoside and the phosphate group. This is achieved using a family of enzymes known as phosphatases. Dephosphorylation can be accomplished with any number of phosphatases.
For this, suppose you have dephosphorylated your vector with CIAP, but now you have to inactivate your CIP and this. For inactivation, you just add equal volume of phenol, chloroform, and isoamyl alcohol to your reaction mixture, and the upper aqueous layer you remove it and transfer it to a fresh tube, and then add sodium acetate and vortex it and add ethanol. So now you got the vector which is CIAP treated, and so it doesn't have phosphate group now and it cannot self ligate to itself. So now the ligation procedure you will be doing, so there is a high probability that your vector will only ligate to the gene of interest or your fragments, not to itself. So the possibility of getting any colony which is false positive or not containing your recombinant this fragments is very less. So like whatever colonies will be getting, there is a high chance that it is positive, having your gene of interest only. Next is purification. Suppose now you have treated the vector with CIAP and you have digested your gene of interest too. So gel purification is a necessary step. As I told that there are these enzymes which work in presence of buffers. So these buffers contain salts to it, and we need to get rid of those salts so that it cannot hamper any further activity, any further reaction. Suppose after digestion you need to ligate, so if those buffers from the digestion reaction should not hamper with the ligation mixture, so you need to gel purify and then column purify your digested products.
DNA ligation is the formation of a covalent bond between adjacent DNA fragments, frequently a vector and a gene of interest. Ligation is usually the final step before transformation in the cloning workflow. DNA ligase may be used to join double-stranded DNA fragments with either blunt or cohesive ends to form recombinant DNA plasmids. A DNA ligase catalyzes the formation of a phosphodiester bond between juxtaposed 5' phosphate and 3' hydroxyl termini in duplex DNA. Ligation proceeds in 3 steps. Initially, the ligase is self-adenylated by reaction with free ATP or NAD. Next, the adenyl group is transferred to the 5' phosphorylated end of the donor strand. Lastly, the formation of the phosphodiester bond proceeds through the reaction of the adenylated donor end with the adjacent 3' hydroxyl acceptor, accompanied by the release of AMP. While T4-DNA ligase is the most commonly used ligase for cloning, there are several options from NEB to choose from, including convenient methods.
So usually we use T for DNA ligase enzyme to ligate your vector and gene of interest. So for this also what we do is we have buffer and enzyme in which it works. So after ligation we transform the product we get, we transform it into bacteria. Like I told you, depending on the, maybe if you use yeast, artificial, maybe you can use this lambda phages and all to transform, but generally we use bacterial cells. So once you got the ligated product, we just say ki this is your recombinant DNA, which is most probably having your vector and your fragments like the gene of interest, and then you transform it into bacterial cells. So for transformation, there are like different methods you can use. First of all, I will be knowing what is the
Lastly,
basis of transformation.
transformation of plasmid means putting a plasmid, which may contain your gene of interest, inside a bacteria. So the process is fairly simple. So the way we do transformation is by creating small pores in the cell wall and the cell membrane by incubating it with divalent cation and a heat shock. So which would create transient pore in the membrane, and that would allow the plasmid to be incorporated inside the bacteria. And later on, if you give enough time for recovery for the bacteria, it would kind of repair their damages and it would grow in the media nicely. And it would also amplify the number of copies of our gene of interest, which is inside the plasmid.
This was the basis of transformation that we create for using different methods, so that your gene of recombinant DNA can go inside and the bacteria can take it up. So for transformation, we can have different method like chemical transformation, electroporation, depending on the bacteria you are using.
In general, two methods of transformation as suggested. One is by chemical transformation, which uses MgCl2 or other divalent cations to prepare chemically competent cells, which are given a heat shock at 42 degrees Celsius for bacteria in general for about 30 seconds to be able to create the pores for plasmid to go inside the cell. And whereas electroporation uses an electroporation buffer, which ensures that cells do not die due to the electric shock, and then you give them a small shock for a very brief period of time, a pulse shock at about 15 kilowatts per centimeter, and for 5 microseconds.
So these video will explain a brief about heat shock and electroporation method of
Transformation
transformation.
is taking the plasmid, which has your gene of interest, and adding that to a new tube. Then we add competent cell. Most of the cases, the competent cell lines are TH5 alpha, and there are many other competent cell lines which are capable of taking the or incorporating the plasmid. Now, after that, we incubate the plasmid and the competent cell mixture for several minutes. Once it is chilled on ice, we quickly put it into a water bath, which is in 42 degrees centigrade, and keep it strictly for 90 seconds. Then we take it out and put it back into the ice. So we have the temperature changing from 4 degrees to 42 degrees centigrade for a brief time point, and then again back to 4 degrees centigrade. So this would create a heat shock. And after 2 minutes of incubating in the ice, what would happen is there would be transient pore in the bacterial cell membrane and the cell wall, and that would allow the plasmid to get inside the bacterial cell. Then now the bacteria are in stress, so they need to grow, and that's why we take the transformant solution and put it into SOC media. Now, SOC media is basically a media which is like enriched in tryptone and a lot of glucose. So this media would allow the bacteria to grow, because if we directly plate it into the plate, so the plate already has some kind of antibiotic resistance, right? Now, in order for the antibiotic resistance to work, the bacteria need to give we need to give the bacteria enough time for these antibiotic resistance protein to be formed, right? So we have to incubate the bacteria for 30 minutes or so. So this would give the bacteria sufficient time to produce the product of the antibiotic resistance gene. Now what we would do is put the bacteria or plate the bacteria into a 10 centimeter plate with a desired antibiotic resistance, let's say ampicillin resistance. So we would put ampicillin in the plate and the bacteria should have the ampicillin, right? Then we would inoculate the colonies and spread the colonies all over the plate. And if we wait overnight and then check, there should be colonies in the plate. And ideally, these colonies should be the transformants, right? But in order to check it, we should take out the colonies, and we can also calculate the transformation efficiency by the following formula that is shown in the screen right now.
So this was about heat shock method. Next, maybe you can use electroporation method.
This pulsating current causes small channels or micropores to be formed in the membranes, through which the molecules in the surrounding area are able to pass.
So basically, electroporation and heat shock, the basis is simple as that. Like we make pores into the bacterial wall so that recombinant plasmid can easily be taken up by the bacteria.
Bit goes on top. Tube goes on top. Chemoid gets placed there. A recovery up there. We're going to check volts 1600. Then we're going to go back to time constant. I'm doing this slowly. I should be doing this a lot faster because otherwise it'll warm up. I'm going to really thoroughly dry off my tube, tap it down, and then put it in this holder. There's a little knob on the side. That knob goes down to that little notch. Throw that in there. Press pulse twice. It'll make a noise, and if we don't hear a pop, then we know it worked. 5.8 is good. We generally want to get above a 5.0. I'm immediately going to stick my tube or my 2IT in there, resuspend it, and this is going to go for recovery. So I'm then going to start to pull it out. So this goes in that.
So using the heat shock method and electroporation, the basis is same, that we want to generate some pores in the wall of bacteria. But in heat shock method, what you do is, by giving heat shock like an ice and at heating it at 42 degree, you generate pores to the wall. And in electroporation, you give an electric pulse and generate pores in the wall of bacteria. Next step is, once you transformed your recombinant DNA into a particular bacteria. Once bacteria multiply, we hope that the plasmid in it will grow into the bacterial cells. So once we have got the colonies on your plate, now you need to screen the particular colony which contains the positive recombinant DNAs. Suppose you might get plasmid which doesn't contain your recombinant DNA, but it will show into colonies, right? Because bacteria is growing. But to check whether this particular bacterial colony has taken up the recombinant DNA, and which gives a this positive colony is there, then you need to screen it. So there are different method of screening. One is blue-white screening.
The transformation efficiency is low, and that is why some of the bacteria has not taken out the plasmid. But this problem could be solved easily. So some of the bacteria have the plasmid, some of the bacteria might not have the plasmid, but the bacteria which has the plasmid has the antibiotic resistance gene as well, right? Now, when you are preparing the media, you're putting antibiotic into the media. So if the bacteria don't have a antibiotic resistance gene, it would die. It would simply die. It cannot survive in that environment. Only if the bacteria has antibiotic resistance gene, it can survive. Without the antibiotic resistance genes, it cannot survive. So this problem is solved. But there is another problem. The second problem is now in the solution there could be two type of plasmids. Like one is just self-ligated plasmid, non-recombinant, and something there could be like a recombinant plasmid. Now, how to understand that the bacteria colony that is formed have the recombinant one, not the self-ligated one. So in order to do this, you need some screenable markers, and very often the screenable marker is present in the form of a particular reporter gene. In this case, the reporter gene is LAGZ. Now, LAGZ is present in the multiple cloning site. Now, if we clone our gene of interest in the multiple cloning site, the LAGZ reporter gene would be kind of perturbed because we are inserting into the locus of LAGZ gene, so its product won't be formed. So LAGZ normally give rise to beta galactosidase. Now, beta galactosidase would convert XGAL into a colored product. So that would make the bacterial colony looking blue. Now, if you insert the gene of interest into that particular LAGZ containing multiple cloning site, then what would happen? Beta galactosidase won't be produced because LAGZ gene is already disrupted. So this is called insertional inactivation. Due to the insertion of your gene of interest, the LAGZ gene is inactivated, and as a result, beta galactosidase is not produced. So the XGAL cannot be cleaved and it cannot produce any color. As a result, the bacterial colonies which have this kind of recombinant plasmid, it would have white. But at the end of the day, there could be two different type of plasmid. So in a plate, you would have some colonies which are blue and some colonies which are white. So remember, the white colonies are those colonies which are recombinant, because in these colonies, the bacteria.
So depending on the vector we are using, it has some screenable markers to it. So usually we use, antibiotic resistance as a selectable marker. Other than that, you can have a vector which has this LAGZ gene and it will give you blue and white colonies. So once your gene of interest is, cloned into that particular or inserted into that particular vector, it will disturb the LAGZ gene. As a result, this beta galactosidase will not be formed, and hence you'll get the white colony. So you can, by looking at the color of colony, you can, screen it out that white colonies are the one which contain the recombinant DNA. Other than that, you can also screen your colonies with colony PCR. So colony PCR is basically, just picking up the colony, and then you just take the genomic. Once you have taken up the colony, you just heat it at 95 degrees so that the cell wall get degenerated, and then you perform a PCR using that colony. And for doing PCR, you need, I'll be showing the video first, then I'll be
cases
explaining.
a control ligation which had no insert to a ligation that should have had all the components. And what you're hoping to see is that your control reaction has nothing and your ligation has a ton of colonies. But more often than not, the reality is that they're going to look really similar, and you're going to have some background, and maybe they'll be twofold, maybe they'll be 1.5-fold as many colonies. So you need a way to find that one colony that worked, and you need it rapidly instead of doing 40 mini preps. So the solution is colony PCR. Now, in order to perform colony PCR, you're going to need 4 things: an epi rack with your PCR master mix, PCR tubes, 1 for each sample, sterile culture tubes containing the growth medium and antibiotic of your choice,
And finally, of course, you're going to need the plates, the bacterial plates that have the colonies that you're going to examine. So the advantage of colony PCR is that you can profile 30, 40, 50, however many colonies you want to pick very rapidly in a couple of hours, instead of having to do an individual mini prep for each colony. So the idea behind colony PCR is that you're going to take one colony and look for your insert of choice. To do this, the first step is going to be picking a colony and transferring it into the PCR tube. So to do this, I use just normal 200 microliter tip, and then you want to swipe one colony. It's very important that you're getting only one colony. Okay, now with the colony on the tip of the pipette, you want to go into the bottom of the PCR tube and basically rub the tip all around the bottom of the PCR tube. You're trying to smear some of that bacteria into the PCR tube. Okay, now once you've sufficiently rubbed the tip into the PCR tube, transferring the bacteria, you're going to take that tip and transfer the tip into one of the sterile culture tubes. These tubes each contain LB, and in my case, ampicillin, because the gene I'm looking for is in an ampicillin-resistant plasmid. Now, one of the most critical parts of colony PCR is being sure that you're careful about labeling tubes. It's very important that the PCR tube is labeled as well as the culture tube, and obviously they correspond. In the end, you're going to be running a gel using the PCR samples and going back to the LB culture tubes to get your sample. So you want to be sure that they actually correspond to each other. All right, so after you eject your tip into the sterile culture tube, you want to go back now to the PCR tubes and add 20 microliters of your master mix. All right, so once you've added all of your master mix to each of the tubes, you're ready for PCR. So after your PCR reaction, you want to analyze each sample by agarose gel. Now, this is going to allow you to rapidly tell which samples had an insert and which samples did not. Looking at the results of the agarose gel, we see our positive controls on the upper and lower half of the gel as compared to all of our 24 colonies that we looked at. What we can see is that we have two positives, sample number 6.
So basically, while performing the colony PCR, you need a specific primer so that suppose your gene of like the primers which is specific to amplify your gene of interest. So you can directly pick up one colony and then perform PCR so that primers can if that particular colony has your plasmid recombinant plasmid, then the gene of interest will get amplified while performing that PCR. Otherwise, if the colony doesn't have recombinant plasmid to it, the PCR will not amplify that particular gene. So you can directly just run it on gel, and if there is a particular product which is amplified, which is your specific to the length of your gene of interest, then you can say that okay, that colony have that colony has plasmid which is having a gene of interest to it. But one precaution you might have to follow while performing this is that you just take you pick up one colony only. I mean, while swiping the colony, it might be possible that you mix two colonies, but that is one thing we need to check in mind, that we are not picking up two colonies together.
Ma'am, I have a question about the last thing you mentioned. So since we are doing genomic DNA library preparation,
Yes.
so what effect, I mean, so here in this case, we are just checking one colony that could have any insert.
Yeah.
So like there is a very little probability that what gene are we looking at, what primers have we designed.
So for that, we go for sequencing. Okay, just a second. I think audio is closed. Can you hear me?
Yes, I can hear you.
So the thing is, there might be a possibility. Sorry, for the genomic DNA library, what you do is you go for sequencing after your cloning. I mean,
Right.
while making fragments, you never know ki this cloning which we are these steps, these are particularly for specific cloning steps only. This doesn't show us that while we are making genomic DNA library, so you will get like thousands of fragments cloned
Right.
into different vector. You'll
Right.
get thousands of colonies. Okay, then you need to just go for sequencing of each of the fragment. But what you can do is you can digest your particular plasmid and see if there is a fallout of that length of your sequence. Suppose you generated fragments of different length है ना. Suppose there is 300 base pair का fragment you have generated. So when you perform the restriction digestion again of this plasmid, no, so you'll get two bands, one of your vector, one is 300 base pair fragment which you have initially ligated. So if there is a fallout, that means that particular colony has some gene of interest to it, है ना?
Okay.
Did you get it, matlab?
So in this, just wanted, like, I've understood the entire thing, but the significance of the last step, which was when we are screening using primers, and we're like amplifying that. So what are those primers amplifying?
Those are in the application where you know the gene of interest. Supposedly, you
Okay.
know one particular gene, and you know these primers can amplify it.
Okay.
But
All right.
suppose if you don't know the gene of interest, but then you just go for either blue white screening, or you
Hmm.
can go for digestion fallout, but not colony PCR. Then you
Okay.
won't be knowing the
Right.
specific primers which can amplify, right?
Okay. Thank you.
Any other question? Anyone else? Okay, then I'll be leaving the presentation now.